All right, let's do it. So in terms of, we're going to start with the genetics. So DNA, deoxyribonucleic acid, function, storage of genetic information. The nucleotides, I'm going to ask you about which ones are purines, which ones are pyrimidines. Okay, that's going to happen. You need to know the components, the nucleoside, which is nitrogenous base and the sugar, nucleotide, which is nitrogenous base, sugar, and phosphate. Backbone, phosphate and sugar, sugar-phosphate backbone. They oriented 5' to 3'. Okay, remember phosphate connects 5 and 3' ends of the carbons of the sugars. Directionality. Since DNA is directional, enzymes can read it in a certain direction, right? So it enables DNA reading enzymes. Structural organization, anti-parallel, double helix. The functions of the minor and major grooves are binding of the proteins. Pairs ATGC, and remember that AT pair is weaker than GC pair because AT pair forms only 2 hydrogen bonds while GC forms 3. Chargaff rule. I want to highlight again that Chargaff rule can be applied only to double-stranded molecules. So if you have double-stranded DNA, Chargaff rule can be applied to double-stranded DNA. If you, hypothetically, have double-stranded RNA, it can be applied to double-stranded RNA. We just don't have the situation. So only double-stranded. Strands are held together by hydrogen bonds, and you can separate them by heating the molecule up, by denaturing it. That makes sense? So that's the process of denaturation. The process is necessary, so only when you split strands apart, you can read the actual sequence of nitrogenous bases. Otherwise, when they are paired, proteins like DNA polymerase or RNA polymerase cannot read the sequence. You need to denature DNA molecule to read it. Difference between deoxyribo and ribonucleotides. So the first difference is that in ribonucleotides you have hydroxyl group OH and the second carbon, 2' carbon in the sugar. And second, in ribonucleotides you will find U instead of T. Remember that. And that presence of hydroxyl group in the second 2' position of the carbon of the sugar makes RNA really unstable. So structural differences: RNA single-stranded mainly, can form intracellular hairpin loop structures, and yes, it can be denatured. You heat it up, hairpin loop structures split. 4 types of RNA. So you need to be able to match the type of RNA and its function, like mRNA messenger. We talked about 4 mRNA, rRNA, tRNA, and microRNA. You have to know the basic function of each of these 4 types. Doesn't go beyond what we talked about in the class. Any questions so far? mRNA, tRNA, rRNA, and microRNA, they are all in the table with all the functions. Relative sizes of genomes. So for relative sizes, just know viruses are the smallest. Then you have bacteria, unicellular eukaryotes, multicellular eukaryotes. Now, be able to identify phenotypical traits and distinguish them from genotype. So if I tell you the presence of the gene, the word gene should immediately tell you that's not phenotype, that's genotype. I tell you bacteria has flagellum or resistant to antibiotics or grows best at, I don't know, 45 degrees Celsius. That's all phenotypical traits. Am I clear? It's observable traits. Okay. Cellular DNA exists in the form of the chromosome, first and foremost. Bacterial chromosomes are circular, and usually there's one. Eukaryotic chromosomes are linear. DNA in the chromosome is bound to histones in eukaryotes and histone-like proteins in prokaryotes. Extrachromosomal DNA in eukaryotes, mitochondria, and chloroplasts. In prokaryotes, it exists in the form of a plasmid. Remember that plasmids are not essential for the cell survival and can be taken out. Does that make sense? So plasmid does not carry essential genes, but it often carries beneficial genes. So in eukaryotes, extrachromosomal DNA, well, it's the DNA of mitochondria and chloroplasts. It encodes several important proteins there. Bacterial plasmids often benefit. We didn't talk about Biddle-Tatum experiment, so don't worry about this part. Transcription. Remember that in prokaryotes, transcription and translation happen almost simultaneously. So essentially, DNA transcription in prokaryotes happens basically in the cytoplasm because there is no physical boundary between the DNA and the cytoplasm in prokaryotes. No nucleus. Does that make sense? In eukaryotes, transcription occurs in the nucleus, behind the nuclear envelope. Now, DNA is the template for transcription, and the product of transcription is RNA. Now, how many strands of the DNA are required? Okay, let me rephrase this. The DNA molecule that is transcribed, can it be single-stranded DNA? No, it cannot. The DNA that is transcribed is always double-stranded, but only one strand is read. Are we clear? Now, 3 stages of transcription are initiation, elongation, and termination. And the process is carried out by the RNA polymerase. Now remember, during the process of transcription, RNA polymerase binds to the site called promoter. Okay, and RNA polymerase reads the DNA during elongation from 3' to 5' end. How many strands of RNA are produced as the result of transcription? Just RNA, just one. So basically it's sort of a rule. So you need double-stranded DNA of which only one strand is going to be read, and transcription will produce single-stranded RNA molecule. We good? Yes.
You said for like the previous question, like how many strands of the template are required in this?
So double-stranded DNA only can be transcribed. But only one strand of this DNA will be read. Did I clarify? Good. Initiation, elongation, and termination. Now, signal for termination of transcription is terminal repeats that force RNA polymerase to stall. Okay? Now, there is an intermediate stage called splicing, which occurs in eukaryotes and viruses. And we talked about alternative splicing. In alternative splicing, different sets of exons are stitched together as the final mRNA transcript. Does that make sense? And alternative splicing allows you to have more than one protein encoded by the same gene. Now, coding fragments are exons, non-coding are introns. Introns are excised, exons are stitched together. This is why we call this process splicing. Now, which DNA occupies more space? Non-coding. And its function is most likely regulatory. Correct. Nucleotides in one codon, 3. One given codon always encodes just one amino acid. But remember this, that same amino acid can be encoded by different codons. Does that make sense? Same amino acid can be encoded by different codons. And we call this feature of human genetic code, the code is degenerate or redundant. The evolutionary advantage of this is that the code is less prone to the mutations. How many different kinds of amino acids form proteins? 21. Okay. Now, translation occurs the ribosomes, which consist of protein and ribosomal RNA, rRNA. When I ask what are the differences between bacterial and eukaryotic ribosomes, I'm not going to ask you to name the actual numbers like 30S and 50S subunits for bacterial. All you need to know is basically they're different. And that's it. Bacterial and eukaryotic ribosomes are different. Amino acids are transported in the cell by tRNA, transport RNA. Okay. Now, order of assembly of translation initiation complex. First, you have small subunit that binds to the Shine-Dalgarno sequence, if it's bacterium, or 5' cap structure, if it's a eukaryote. Then, mRNA and small subunit, they collectively bind the first tRNA, the methionine tRNA, AUC codon. And then, finally, the large subunit joins the complex. So the order will be 30S plus mRNA plus tRNA plus 50S subunit. Okay, so small eukaryotic subunit, as I just mentioned, binds to 5' cap. 5' cap structure. Prokaryotic binds to Shine-Dalgarno sequence. Now, first amino acid, thionine, first codon, AUG, protein synthesis happened. Oh, okay. So this is really important. So in prokaryotes, remember we talked about continuity of transcription and translation in bacteria. when you have DNA with the DNA polymerase on it, which produces RNA molecule, which has ribosomes on it, and each ribosome produces a protein molecule. Remember that picture. So you have to understand that since there is no boundary between transcription and translation in bacteria, there is no splicing. Translation immediately follows and occurs almost at the same time with the transcription. Is that understood? Translation occurs almost at the same time with the transcription in bacteria. In eukaryotes, where there is nucleus, transcription happens first, then splicing happens, RNA modification happens, and then only translation occurs. So in eukaryotes, transcription and translation are distinctly separated. Are we clear? Does that make sense? So protein synthesis always happens on the ribosomes, but say in eukaryotes, ribosomes can be on the membrane in the endoplasmic reticulum. In prokaryotes, ribosomes are in the cytoplasm, always. A site, incoming tRNA binds to it. P site, that's where the tRNA with a growing polypeptide chain is found. And E site is the site of exit for the tRNA. Now, what is transferred to what? Polypeptide chain is transferred to the incoming amino acid. Ribosomes stop when they encounter the stop codon. What happens if there is a selenocysteine insertion sequence? Then ribosome keeps moving, and instead of stopping, inserts selenocysteine amino acid. And it's going to be moving till the next stop codon. So main differences between pro and eukaryotic gene expression. So first of all, bacteria and eukaryotes have different ribosomes. In bacteria, there is no splicing. In eukaryotes, there is splicing. In bacteria, there is no nuclear membrane. In eukaryotes, there is nuclear membrane. And this means that in bacteria, translation and transcription happen practically at the same time. In eukaryotes, transcription and translation are separated both in time and in space. Finally, in eukaryotes, each gene is transcribed as the single mRNA, and we call this monocystronic RNA. In bacteria, several genes can be transcribed as the large mRNA molecule, and we call such mRNA molecule polycystronic. Are we clear? So we just talked about spatial and temporal restrictions, and I refer to, presence of nuclear membrane, that's spatial, and temporal. RNA has to be modified, RNA has to be spliced. Mono and polycystronic, just discussed them. 3 subfamilies of the Herpesvirinae: alpha, beta, and gamma. Herpesviruses are enveloped. They have double-stranded DNA genome. Tegument contains the proteins necessary for the early onset of infection. Well, I kind of gave you the order. Immediate early genes are expressed first, then early genes, then late genes. Herpesvirus DNA is replicated in the nucleus, and herpesvirus DNA is not incorporated into the cellular DNA. When herpesvirus becomes latent, meaning it doesn't express genes, it doesn't replicate. Its DNA exists in an episomal form in the nucleus. Episomal means above the chromosome, right? So herpesvirus DNA is not a part of the chromosome. Herpes simplex virus, they establish latency in neurons. Cytomegalovirus establishes latency in the monocytes. And Epstein-Barr virus establishes latency in the B cells. Now, herpes simplex 1 and 2, they cause herpes. Herpesvirus 1 establishes labial herpes, and HSV2 causes genital herpes. These viruses can be transmitted via bodily fluids and vertically, and there is no vaccine. Prevalence of herpes simplex increases with age, and HSV1, the labial herpes, is more prevalent, meaning that it infects more people. Does that make sense so far? HSV can cause encephalitis in immunocompromised and a bunch of other diseases. I really want you to focus on encephalitis. In newborns, all you need to know is that congenital HSV is extremely severe. Acyclovir is used to treat herpesvirus infection. Varicella zoster causes chickenpox and shingles. Virus is transmitted with the bodily fluids and respiratory, and yes, there is a vaccine. Cytomegalovirus doesn't really cause any serious disease, okay? So, not really, no. Virus is transmitted bodily fluids, there is no vaccine. Now, I'm going to digress for a second. You really have to appreciate the commonalities between herpes viruses. They can all become, so let's see, they all have DNA genomes. They can all become latent. They all can be transmitted via bodily fluids, and they all are enveloped. Does that make sense? That's really a common theme across all of them. CMV can cause a variety of diseases in immunocompromised, from keratitis to pharyngitis to liver damage. In newborns, CMV infection in newborns is extremely severe. Treatment options, or drugs, you can focus on acyclovir again. Epstein-Barr virus causes infectious mononucleosis. It's transmitted by bodily fluids. There's no vaccine. It can progress to develop two types of cancers: Burkitt's lymphoma and nasopharyngeal carcinoma. B cells are latently infected by EBV. Kaposi sarcoma herpes virus, guess what? Causes Kaposi sarcoma. Skin disease, virus is transmitted bodily fluids, no vaccine. So common features we just mentioned. We're good? Do we need to highlight them again, or are you fine? Fine? They were mentioned right here. So that's your common features. Purpose of DNA replication, multiply DNA to transfer to the next generation. At which step of the cell life cycle, I'm not asking this for eukaryotes. Where does it happen in eukaryotes? In the nucleus. 2 strands of DNA backup, semi-conservative. Each new DNA molecule consists of one old and one new strand. Now, functions of the enzymes. I'm not gonna go over it again, okay? But you need to know the function of the main enzymes that we have in the table that we talked about. Am I clear? Direction 3 to 5 prime, reading. Replication start at the origin, AT pair is more abundant. So, okay, supercoiling. I'm not going to mention any of the enzymes. Supercoiling when DNA is tangled. Unzipping is separation of two strands. Primers are short RNA sequences that are necessary to mark the starting point for DNA polymerase 3. Leading strand is synthesized in the continuous fashion. Lagging strand is synthesized in the form of Okazaki fragments. Okay? They get stitched together by ligase. They formed on the lagging strand. Why are they formed? Because DNA polymerase on the lagging strand has to move in the direction opposite to the direction of replication fork. Okay. Concatenated chromosomes. Chromosomes connected like two elements of the chainmail. They're separated by topoisomerase 4. Mutations, changes in DNA. Protein cannot be mutated. It can only be changed. What can cause a mutation? So know that it can be a mistake in the replication, or carcinogens, mutagens can cause it. Types of mutations. Know the 3 types for point mutations and the general idea of frame shift. Okay? So, you will have to match the description and the mutation. Nitrous acid nucleoside analogs. All you need to know, they cause point mutation. Intercalating agents. They stick themselves between the nucleotide pairs. Ionizing radiation. Double-strand breaks. Non-ionizing radiation. Thymine dimers. They can cause either point or frame shift. Excision repair. Remember we talked about excision repair with thymine dimers. No excision repair and no photoactivation repair. Just, we don't have time to go over it again. Ames test. There will be one question on the exam on the Ames test. Okay, so basically it will address what happens when you expose oxytrophic mutant to a mutagen. What kind of growth you're going to see in the end. Remember we talked about it? When you expose an oxytrophic mutant to a mutagen, then you're going to see growth on, say, nutrient deficient medium. If it's not mutagen, there will be no growth. So review, please review the Ames test. If we will go into it, we will spend another like 20 minutes going over. But one question is going to be long, the answers are going to be long, so expect it. Understand the Ames test well. Okay? We call the organism that return to the original genotype revertants. Oxytrophic mutant is the mutant that requires a certain nutrient. 3 mechanisms. So, match, conjugation, transformation, and transduction with a description of it. Am I clear? There will be questions. Blah, 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 blah is, and you have to tell whether it's transformation, transduction, or conjugation. You have to be able to clearly match it. Know the difference between specialized and generalized transduction at the level of. Specialized transduction is carried out by lysogenic phage, generalized by lytic. Bacterial, I'm not going to ask about bacterial transformation in biotechnology application. Importance of bacterial transformation for public health. All of these horizontal gene transfers contribute to the spread of antibiotic resistance. Diversity of eukaryotic organisms is increased by sexual reproduction. And prokaryotes cannot do sexual reproduction. This is why horizontal gene transport. Okay. In HFR, I'm skipping this because this is basically a description. So if you answer those questions, the detailed description. So transformation, obtaining free DNA from the environment. Transduction carried out by bacteriophages. And conjugation is exchange of fertility plasmid between two live bacterial cells. In HFR cells, fertility plasmid can incorporate itself into the chromosome and go back. Now for operons, know what structural genes are: operator, promoter sequence. You should be able to answer the question, bless you, like this. What binds to the regulator? Repressor molecule. You also should be able to recognize the mechanism of, if I give you a description of the operon, you have to be able to recognize if it's inducible or repressible. You have to know, in terms of the mechanism, that in inducible operon, the molecule, the substrate of the enzymatic reaction, binds to the repressor and allows transcription to occur. In repressible operon, the product of enzymatic reaction binds to the repressor and stops transcription. Riboswitch is the RNA structure that regulates either transcription or translation. Riboswitch can be turned off by binding of a small molecule. Okay? Its central part is hairpin loop. Now, metabolism, catabolism, anabolism. People, definition. This. Remember the table with heterotrophs versus autotrophs. You know, chemotrophs versus phototrophs. You saw the questions on today's quiz. Something in that light. Okay? So, chemoautotroph, what's the source of energy? What's the source of carbon? Oxidation reduction. There will be questions with a lot of like, today's quiz, I will use it as an example. You had questions, what is oxidized for, right? Or there were some answers where it says NADH is reduced or NAD is oxidized. Neither of these processes is possible. NADH cannot be reduced because it already is reduced. And NAD cannot be oxidized because it is oxidized at this point. Does that make sense? If you will ask me what is oxidation and what is reduction, I will not answer. Just upfront, okay? I will not answer in terms of the electron transfer. I won't be able to help you with this. You have to be comfortable with this terminology, which I will be using in asking questions. Am I clear? Okay. So know the electron transporters, NADH and FADH2, NADPH for plants. Know the reduced and oxidized form. Remember, the reduced form always have more hydrogens. Like NADH is reduced, NAD is oxidized. FADH2 is reduced, FAD is oxidized. Am I clear? Exergonic, endergonic. Exergonic releases energy, endergonic consumes energy. Anabolic, catabolic, already talked about it. This, just know it, ATP synthesis, say anabolic, endergonic. For enzymes, know the features, very specific and not wasted in the reaction. Increase rate but not ratio. Usually proteins, less RNA. Okay, we got this, Know what the substrate and product. Be comfortable with the terms like substrate binds to the active site, not the product. In the active site, activation energy decreases. Reaction conditions change, and the performance of the enzyme can change as well. That's, Victoria, to your question today. The picture that I had, that enzyme works in the range of conditions. Okay. A constitutive regulated, produced constantly regulated endo-exo enzymes, inside outside. Competition. Competitive inhibitor binds to the active site, non-competitive to the allosteric side. Allosteric activation, activator binds to some other side, allosteric side. Does that make sense? So remember that non-competitive and allosteric inhibition are synonyms. Enzymes usually regulated by a negative feedback mechanism. Glycolysis. So start with glucose. Intermediate is G3P, glyceraldehyde 3-phosphate, which is converted into pyruvate. If oxygen is absent, pyruvate will be fermented. If it's present, pyruvate will enter the transition step. During glycolysis, you have 2 ATP molecules that are produced, 2 NADH, no CO2. Pyruvate is converted to acetyl-CoA during transition stage, and this molecule enters the Krebs cycle. It contains 2 carbons from the original glucose. Acetyl-CoA combines with oxaloacetate, producing citric acid. So basically we're doing Krebs cycle. Now we have 2 plus 4 equals 6. Citric acid is converted to alpha-ketoglutaric acid. So we have 5 carbons now. 1 carbon is lost in the form of CO2, and NADH molecule is produced. Alpha-ketoglutaric acid is converted to succinyl-CoA. Another CO2 is formed, and NADH is produced. So for Krebs cycle, you don't need to know anything more than I just told you, but you must know the products of the Krebs cycle. 8 NADH, 2 ATP, 6 CO2, 2 FADH2. Does that make sense? Per molecule of glucose. Now, NADH and FADH2 carry electrons to electron transport chain. Original glucose molecule is completely oxidized during Krebs. So you need 2 Krebs cycles to completely oxidize 1 glucose. So we got this. Summary outcome of the transitional and the Krebs. We didn't talk about this too, don't worry. So electron transport chain. Energy of electrons is used to pump hydrogens across the membrane, creating electrochemical gradient. Okay? Hydrogens cannot cross the membrane directly, so they cross the membrane through the ATP synthase, carrying out, and the energy of proton movement, proton diffusion, is used for ATP synthesis. Okay? Organisms cannot carry out aerobic respiration either due to the lack of oxygen or lack of the genes encoding electron transport chain. Fermentation occurs without oxygen. Final electron acceptor in fermentation, organic molecule. Pyruvate in lactic fermentation, acetaldehyde in alcoholic fermentation. You need to know this too, and that's it. In anaerobic respiration, inorganic molecule, nitrate, sulfate, carbonate. ETC in aerobic and electron transport chain in aerobic and anaerobic respiration are different. Fermenting organisms lack ATC. Reactive oxygen species are formed during the respiration, and they're neutralized by enzymes. Fermenting organisms lack these enzymes. They don't have them. They cannot neutralize reactive oxygen species. This is why they can't respire. Fermentation, lactic and alcoholic, okay? Lactic fermentation, fine electron acceptor pyruvate, alcoholic acetaldehyde. Chain, positive, strep pyogenes, causes impetigo. Streptolysin breaks red blood cells. Can be detected using blood agar. Complications. Know about scarlet fever and rheumatic fever, okay? Joints and heart. Transmission route, respiratory, no vaccine. Carriers, transmission route, it's in the microbiome. Metabolism of strep mutans, it's fermenting. It ferments fructose producing acid. We clear? Strep pneumonia causes pneumonia and adult community-acquired meningitis. Transmission route respiratory, there is a vaccine. Strep A. galactiae causes neonatal meningitis, and it is commonly found in the vaginal microbiome. Now this one, remember the flow chart that I had for proteins, lipids, and carbohydrates. So proteins, eventual products of lipid metabolism. Glycerol and acetyl-CoA. Glycerol enters glycolysis, becoming glyceraldehyde 3-phosphate. Acetyl-CoA enters Krebs cycle. Acetyl-CoA is the product of beta oxidation. Proteins, amino acids, are going through deamination and transamination, and they can either be converted to pyruvate or become intermediates of the Krebs cycle. Regarding photosynthesis, light-dependent and light-independent reactions for the pigments allow to absorb different wavelengths of light. Light-dependent stage gives you ATP and NADPH. Light energy is collected at the light harvesting centers. Energy is directed at the reaction center, where electron is energized. Okay? Electron donor will be water. Okay? Oxygen of water is oxidized, becoming molecular oxygen. Electron is energized and enters the electron transport chain, okay, which then uses energy of that electron to pump hydrogens across the membrane. Diffusion of hydrogens back leads to the ATP synthesis. Now, water oxygen is oxidized. Reaction center in photosystem 2 compensates for the lost electron by oxidizing oxygen. Can other molecules be electron donor? Yes. If photosynthesis produces no oxygen but something else, we call it anoxygenic photosynthesis. After photosystem 2, electron enters photosystem 1, gets energized again, and reduces NADP to NADPH. Okay? In light-independent reaction, it's Calvin-Benson cycle. Carbon dioxide interacts with ribulose biphosphate. Remember that story? Interacts with ribulose biphosphate, producing, after several steps, 3-carbon intermediate, which all boils down to G3P, glyceraldehyde 3-phosphate, which, as a part of Calvin-Benson cycle, is used to synthesize molecules like glucose and fructose. What you need to know that carbon dioxide in Calvin-Benson is being fixed to produce organics, and ATP and NADPH are used in Calvin-Benson cycle. Okay?